Let's study the second minor metabolism, that is the biosynthesis of cholesterol. All the carbon atoms of cholesterol are derived from the acetyl coenzyme. The major sites of synthesis of cholesterol are liver, adrenal cortex, testes, ovaries, and the intestine. The steps are six steps for the cholesterol biosynthesis. First is from the acetoacetyl coenzyme synthesize. The coenzyme is removed and the HMG coenzyme synthesis. The enzyme forms the beta hydroxy beta methyl glutathione coenzyme. The third step is HMC coenzyme reductase, that are rate limiting for the formation of mevalonate. The kinase takes place in the fourth step and the 5-phosphomevalate turns to the 5-pyrophosphomevalonate. Again, the kinase takes place and the decarboxylase takes place for the formation of isomerase dimethyl pyrophosphatase. Then the transferase, and the squirrel synthesis and the epoxidase. The sixth step is cyclase, then the zymosterol, desmosterol, and the cholesterol is the last step for this 27 carbon atom is formed. Yeah, some names of the sterols, that is the cholesterol, bile acids, glucocorticoids, and the mineralocorticoids, the testosterone, estrogens. The most important, the significance or the use of some steroids are given below. These are the some functions of the cholesterol, that is the cell. It is used in the cell membrane, the nerve conduction, and the bile acids and the bile salts for the absorption of the fat absorption. The steroid hormones are used. The vitamin D3 esterification. These are the some functions of the cholesterols. There are three types of lipoproteins: very low density lipoproteins, and the high density lipoproteins. Let's study the very low density are synthesized in liver from glycerol and the fatty acids and the hepatic cholesterols. The half life of VLDL in serum is only 1 to 3 hours. In peripheral tissue, it is taken up to adipose tissue and the muscles. The function is to carry the triglycerides from the liver to peripheral tissue for energy needs. The given diagram is of the low density lipoproteins, that it contains the ApoB100 on the top and the cholesterol polypeptides. It is made up of phospholipids and triglycerides. The low density lipoproteins are the transports cholesterol from liver to peripheral tissue. The half life is 2 days. The low density lipoproteins taken up by the peripheral tissue by receptor mediated endocytosis. The main function of low density lipoproteins are the steroid hormones and the incorporative in the muscles. It is how the low density lipoproteins are digested from with the use of lysosomes. The cholesterol deposited. The high density lipoproteins transports the cholesterol from peripheral tissue to liver. The metabolism and the function in the metabolism are seen that the liver it goes to intestine from the heart and blood vessel it goes for the maturation cycle. This is the overall summary for the lipoproteins metabolism from the very low density lipoproteins and the high density lipoproteins together work in the muscle cells also to the peripheral area surface, how it is worked from the liver, intestine and the blood vessels. It is shown in this single diagram. Formation of bile salts is the main precursor of the formation of bile salts are the cholesterol. The cholesterols are divided into cholesterol, then the chloric acid, and the chloride coenzyme, then